welcome you as we showcase what our beautiful ladies are doing in Lagos, Nigeria. Today, first of all, we'd like to share with you the history of Soapbox science all over the world. This is a public speaking event that was initiated by two wonderful scientists, lady scientists, Serian Sumner and Nathalie Petrelli. They started this event in London in 2011. This year, I can assure you there are over 45 events worldwide in 15 countries, no less. And this is one of the two events in Nigeria this year. So this Soapbox Science, what is it really? It's a public speaking event that promotes women who are working in science to share the research that they do with the public. Systems, no theater, no lecture theater, no projector, nothing. They just come and stand traditionally on the soapbox and then speak to the public in the open space. But because of COVID-19, as we all know, we can't have such public events. That's why we're coming to you again virtually this year. This is the third event that Soapbox Science League in bringing Soapbox science to you. Now, the team is made up of some very beautiful scientists led by Dr. Orode, who, well, as I said, is the lead of a team. Can you hear me or am I frozen?
We can hear you. Are you sharing your slides? I can't see anything at the moment.
I am. Can you see my slides?
Not at the moment. Do you want to stop and just start again and let's see whether we can see it?
Okay. All right, I'll do that because I was sharing. Let me stop the share and then do a new share. It's as though I'm frozen. I can't get anything done. But I'm glad you can still hear me.
Yes, we can. We can.
So what do? I'm frozen. Nothing is getting done from here.
Okay, just carry on. Just carry on.
Anyway, okay, all right. So the team, Dr. Orode, pharmacist Flora, Tochukwu, that's Dr. Tochukwu, who is one of our technical persons. And then we have, yes, that's my slide. Can you see it now? Then there is Dr. Oyinkan Adeshaki, who shares the job of logistics with me. I'm Simiat Elias. There's Elsie, our finance manager, and of course, the pharmacist Flora is our partnerships manager. So I hope you can see the slides now, but if you can't, I'm glad that you can hear me. This year we have, originally we have 7 speakers, and they will be speaking on the work that they do in the lab, bringing it to you, breaking it down to you, and encourage the next generation of female scientists. We have Moyinoluwa Akintunde from the University of Ibadan, who will be talking about the making of useful products by bacteria when they use farm waste as food. Dr. Rebecca Chukwuanukwu will be talking from Nnamdi Azikiwe University about afraid of getting vaccinated against COVID, understanding your...
Okay. While we wait for Dr. Simia to reconnect, can you just tell us in the chat, say hello in the chat and tell us where you're joining us from today, so we can see who's represented here. Just tell me in the chat where you're joining us from and say hello to us. Can everyone hear me? Oh, yes. Hello. Thanks, Jane, joining from Ikeja. Thank you. We have 08 joining from London. Thank you very much. We have Ume Francis from Uniben. Great. Great. Thank you for joining us today. Where's my screen gone? Here we go. Who else? Well, I'm here in the UK. Hello, hello. It's getting cold here. Winter is upon us. Oh, we have Kenny from Uniben as well. Ooh, Africa is represented today. We have someone from Burkina Faso. Amazing, amazing. That's also, I believe, is from Burkina Faso. Yes. Redmi is joining us from Benin. And Dorothy from Unilag. Of course, Unilag has to be represented here today very well. Then we also have, somebody from Virginia, USA. Arthur is the one from Burkina Faso. Fabulous. Thanks for joining us today. You are in for some very interesting talks today. Okpayemi from Uniben. Uniben, wow, there's a lot of you here today. Well done, well done. Thanks for joining. Thanks for joining us. We're also streaming live on YouTube and Facebook. If you can reach out to other friends or families that might want to join us as well, they can watch the live stream on YouTube and on our Facebook page at Soapbox Science Lagos. Alrighty. Now, I think Dr. Simiat might have to do the rest of her presentation later on. And let's start and let's get to know some of our speakers today. So I am just going to spotlight speakers for everyone, and then I'm going to do a get to know you session with them. So let me just get all of them together. Speakers, please have your video on, or else I won't be able to spotlight you, and our audience need to see you. Okay, got Professor Herrera there. Then we have Ifeoluwa, and then we also have. Hold on a second. That's wrong. Okay. Yes, we have Dr. Oyelowo. Here we go. And then just two more to go. Speakers, please put your video on, or else I won't be able to spotlight you. Thank you. Fabulous. Okay, so yes, speakers, please unmute yourselves. And I'm going to throw random questions at you so our audience can get to know you. So I'm going to go first to Professor Herrera. Tell us what you do, introduce yourself and your career so far. Now, let's remember time limits. 1 minute, please. Thank you.
Thank you. My name is Akhere Omonkhua. I'm a teacher at the University of Benin. I teach medical biochemistry. My main area of research is medicinal plant research, where I look at medicinal plants that treat diabetes, but I also collaborate with colleagues to look at medicinal plants used to treat malaria and reproductive disorders. I'm currently the head of my department, and I also am the project coordinator of a World Bank assisted project here at the University of Benin. I think the major thing about me is that I love teaching. I think my world revolves around teaching and research and my students and of course my family. Thank you.
Amazing, thank you for that. Okay, Dr. Moyinoluwa, tell us about your career. What do you do?
I just concluded my PhD in industrial microbiology.
Wow, congratulations.
Thank you. So it's been an amazing journey so far, doing research in microbiology generally and moving my research from basic microbiology into an interdisciplinary research, having polymers, microbial polymers, and microbial physiology, combining both together.
Excellent, excellent. Thank you. Ifeoluwa, same question for you. Oh. Please unmute yourself.
Okay, yeah, good afternoon, everybody.
Good afternoon.
I'm Ifeoluwa. I currently work as a research scientist at EHA Clinics at Abuja. And my main area of research are parasitic and infectious diseases generally. I've been involved in quite a number of researches, and I'm a researcher, a parasitologist by training. Thank you.
Excellent, excellent. Okay, so next question. Dr. Rebecca, are you there? Your video went off. I don't know if you're having internet issues. If not, I'll just move on. Okay, next question to Oluwakemi. What attracted you to STEM?
Thank you very much. What attracted me to STEM was my journey from the secondary school. I was fascinated by the subject biology because it deals with plants, animals, and humans and how it relates to the environment. And I also had practical classes that made me, made things real. Practical lessons, practical classes, and I also had great science teachers. So that attracted me to biology as a course.
Fabulous. So it was right from school?
Yes, right from secondary school. Yes. So that attracted me to take on a course, relating to living things and their functions. Yes.
You must have had good teachers then.
Yes, I did. I mentioned that. They were great teachers.
Good teachers like Prof. Akhere. Prof. Akhere, what attracted you to STEM?
I think that right from the beginning, I've always been a curious person. And that's carried me through primary school, secondary school. But I think that my love for science was really ignited in junior secondary school, GSS 3 specifically. I had a wonderful chemistry teacher, Mr. O.T. Jonah. I have been trying to get in contact with him because I think he had a marvelous impact in my life. And I think one of the things he did was to bring science to life for me. Let me put it like that. Because sometimes when we look at science, it's very abstract, very out there. But when you have a teacher who brings it down to you and you see it's real, it makes a difference. And he did that for me. And I think he started my journey on this road of science. And so when I came into the university, I was blessed with many more teachers who had that passion. So I think that one of the greatest respect I have is for teachers. Perhaps that's why I wanted to be a teacher.
Maybe,
even though we're not paid so well, but at least we're making an impact.
Absolutely, absolutely.
Yeah,
okay. Rebecca, the same question to you. So what attracted you to STEM?
My attraction to STEM had to do with getting to know, seeing things for myself. I was so interested when I heard about certain things, read about them, I wanted to see them for myself. And when I heard about the microscope, something that could make things really many times over, you could see things that you could not see with the naked eye, I was very, very attracted. So I moved into it. So that is how my journey for STEM started, want to see it for myself, want to provide the solution, be part of the solution, understand. And then I also love teaching. So I like to break it down to my students in the way that it's literally unforgettable. So that is how I got into it. And I had very good teachers as well.
Yeah, teachers are very important. They, they help to instill the passion. Yeah. Ifeoluwa, same question to you. What attracted you to STEM? Remember, we have lots of young girls that might be listening. So they'll be what attracted you to STEM?
Okay, so what attracted me to STEM basically was my teacher, my biology teacher. Even though I think when I was in SS1, biology would seem very difficult, it seemed very, very bulky. When you have a math note of maybe 60, you won't even use it. You won't exhaust it. When it comes to biology, it seems like you're joining 4 notebooks together, and you have a teacher that can break it down to the simplest form. So I think my teacher was the one that actually brought that on. And I think my level of curiosity also helped me because I usually want to know. When I'm told something, I want to know what it is. So I think it was my curiosity that also led me to STEM.
Fabulous. Great. Okay, so Dr. Moyinoluwa, and then Oluwakemi, you can answer after her. What attracted you to STEM?
Okay, what attracted me to STEM was biology, basically. I liked biology. In fact, I love biology. And my biology teacher was very good. But actually, my teacher that attracted me to science basically was Miss Ajaro, and she was in chemistry. But I didn't like chemistry. I liked only biology, and I always wanted to study pure and applied biology. So I didn't want any other science but biology. So it was the attraction to she taught me biology also, and she made me like it. And it turned out that she made me like chemistry, but I didn't like chemistry until I got to the part of science, or I got maybe, let's say, master's, and my research had to do with chemistry. And it turned out that chemistry was not so bad. Miss Ajaro made me like chemistry somewhat. It was not too difficult. And so from there, it was like, oh, biology and chemistry, I can combine it. And it was fun, actually. It's been fun because that memory of chemistry was very difficult. I liked only biology, but I found myself doing biology and chemistry and microbiology. So it's like, so indeed, it's not that bad, because this woman impacted my life in a way that I probably didn't think of then, but I'm looking at it now and I'm saying, really, it was not that bad. And she really tried, because till now, I try to get in touch with her once in a while, but I always remember her when I remember my journey in science.
Fantastic. Chemistry is awesome. How can you not like chemistry? Chemistry is fab. Okay, so Oluwakemi, what attracted you to STEM?
Okay, like I already said, that biology is about plants and animals and human beings and their functions, in the environment. One thing I would like to add is that my teachers encouraged me to, to take a course in the sciences. So I think guidance and counseling could also help you, or could also make you make choices that at the end of the day you may not necessarily regret, because I don't regret being in science.
Fantastic. Thank you so much. Thank you so much. So before we move on, and I just want you to answer in 10 seconds. In one word, describe your relationship with science. One word. In 1 second even. Professor Akhere?
Unraveling.
Okay. Ifeoluwa?
Fabulous.
Oluwakemi?
Exciting.
Yes. Okay. Moyinoluwa?
Interested.
And Rebecca? Rebecca? Okay. So we've had those exciting, fabulous, intriguing, unraveling descriptions of science. And now?
Solutions.
Okay. Sorry?
Solutions.
Somebody said solutions. Okay.
Solutions. Yeah.
Fabulous. Thank
Rebecca.
you. Thank you. Excellent. So we've got to know a bit about our speakers today. Dr. Elias is back, so we'll just give her a few minutes to finish what she was saying. Welcome back, Dr. Elias.
Yes. Okay, so I can speak now. Do you still want me to share my screen?
Yes, just carry on with the last thing you were going to say. Yes, please. I'm just going to make you co-host so you can share.
You need to allow me. Yes, you need to allow me, please.
Yeah, you can do that now.
Okay.
Go ahead.
So yes, thank you. Just a minute. I'm sorry, you know what happens with network sometimes. Just locked me all out. I couldn't get back in. So I was talking about speakers for this year. The next person that was going to introduce is Dr. Oluwakemi Oyelowo from the University of Lagos, who will be speaking to us on the role of some edible seeds in human reproduction. Wow. So when you eat seeds, may actually be helping your reproduction. That's what she's going to tell us. I don't know, but I hope to learn something. Then Dr. Olubunmi Shoraibi from Lagos State University will be speaking on the role
Oh, internet, please be kind to us. Okay. Let's move on. And let's get the show on the road. Right. So we're going to have our first speaker come up now. And
also that, I think that's it. And then to show our sponsors. That's the last thing I would like to do if my slides would just cooperate. Yes, so encapsulate, we thank you. Dish, we are honored that you're willing to sponsor us. Elias and Associates, we appreciate your help with promoting Nigerian women in science. And that's a lovely picture of all our speakers. Thank you and welcome again. Have a lovely, lovely time. I'm sure you're going to have lots of fun. Thank you.
Thank you so much, Dr. Elias. And yes, as I was saying, yes, we'll move on to our first speaker now. If you just stop sharing the screen for me.
Okay.
Yes, here we go. All right, so our first speaker today is now Dr. Moyinoluwa Akintunde. She has a bachelor's degree in microbiology from Covenant University, a master's degree in industrial microbiology from the University of Ibadan, where she recently obtained her PhD. Congratulations once again. Her research was focused on the use of agro wastes as substrates for production of value-added products by microorganisms. She did part of the research at the Swedish Center for Resource Recovery, where she worked on bacteria, cellulose production, and also bioplastic production. Over to you, Dr. Moyinoluwa. Have you got your video on? Fantastic. Dr. Elias, please stop sharing. Thank you. And here we go. Over to you.
Good afternoon, everybody. So I would like to start by asking us some questions. And the first question I would like to ask us is, what do we do with our watermelon bark when we eat the juicy, succulent part of our watermelon? Or after peeling our yams, what do we do with the peel or the potato peel after peeling our potatoes? Or what do we even do with our corn cob when we take off the grains from the cob? Most of the time we trash them, we throw them away. Sometimes I like to say that sometimes the goats we throw them to are even tired of eating these things sometimes, because they're always just everywhere and they've had enough and they just want the others to just go. So these things constitute nuisance in our environment. But do we know that those same leftovers or trash are food to some other kind of living organisms? And those living organisms need them on a regular basis. So example of those living organisms are bugs. Most of the time when we think of bugs, when we say bacteria, germs, bugs. And this is the first thing that when I want to see a bug, when somebody says, this is a bug, it's ugly, it's not looking good. But do you know that these bugs can eat these things as food for their own growth and their own metabolism to produce things that we need? So, and I would like to say that we have good bugs and we have bad bugs, meaning not all bugs are bad. There are some bugs that they are part of our daily life. They are part of what we eat every day. So, for instance, in your yogurt, if the bugs don't take part in the yogurt, you can't have yogurt. If they don't take part in cheese, you can't have cheese. If they don't take part in bread, you can't have bread. So example of those good bugs, we have something we call Lactobacillus. I want us to, every time you buy yogurt, look behind the pack and you will see starter culture. You will see that thing that they write the name. It may look strange, Lactobacillus. Yes, it's a starter culture used to make yogurt, used for cheese. Also in our bread, what makes our bread rise or what makes puff rise? We have baker's yeast, Saccharomyces cerevisiae. So we use these things on a daily basis. So these bugs enhance the flavor, the taste, the texture of this product and this produce every day. So now, what are the other important things that bugs can make for us? Example is cellulose, and it brings you to my research. When we think of cellulose, we think of trees, plants. But do you know that this same cellulose from trees is very important in our daily life? Drugs formulation, to make drugs, they use cellulose. To make paper, toilet roll, we use cellulose. To make gelling materials or thickening materials for food or for paints and some other things, this cellulose is also useful. So we see that cellulose is useful in our daily life. But when we think of cellulose, do we think of cutting down trees all the time? So to get cellulose from a tree actually is actually quite a long process. And I want us to see this process. I don't know if we can see it properly. It's a process that requires the use of machines. It requires the use of chemicals. It requires the use of different things that are not too good to our environment. So when we use the cellulose that are these machines and we release chemicals that are not too good to the environment, we have effect on our climate, negative effect. So how do we deal with these things? We can get cellulose from bacteria, yes. But when we say our climate is also important to us. So for instance, why don't we need to cut down our trees? It's a sunny day today, and I'm coming from the market and I'm working in my estate and it's so sunny, and I get to a place where there are so much trees and I just stay there because it's cool. The environment cools me down. And I'm wondering, why is the tree important? But I was just walking some minutes behind and it was so hot. Why is it cool here? And yeah, the trees are cooling because the trees have cooling effects. They have a way of stopping the sunlight directly from hitting the houses and hitting the floor that is hot and our roofs that is hot. So these trees are important to us. Cutting down these trees for making of cellulose or to use cellulose to make a lot of things is not the best for us, so we need to preserve them. So I'm saying that an alternative to that tree cellulose is bacterial cellulose. Bacterial cellulose is very straightforward to get. All I need to do is get a bacteria that will make cellulose, or a bug, instead of using the word bacteria, a bug that will make cellulose to us. And that bug is everywhere. I can get it from my rotten fruits, that there's a heap of rotten fruits in the market. I'll get my cellulose making bug from there because the rotten fruit harbors it. And all I need to do is get my farm waste, my peels, my corn cob, my watermelon bark, grind them, put them in a liquid and put the bugs in it. And the bugs make my cellulose for me in a very straightforward process. So I would like us to look at this and see, my bug will make the cellulose. And the cellulose picture is not so clear. The bugs will make the cellulose, and the cellulose is in the medium. And after that, I would have my cellulose out of the medium in a very straightforward process, because the bugs will feed on that corn cob, it will feed on that watermelon bark, it will feed on that potato peel to make and it is very easy, straightforward. I don't need to go through any chemical process. All I need to do is remove my cellulose and wash it, and I'll have something like this, a very nice film that I can use, and I can use it as a paper like this and write my name on it. I didn't do anything to it. I just dried my cellulose and I wrote on it. I can make it into a color and cut it into smaller pieces. This is made into purple. How did I dye it? I used just normal food coloring we used to bake and design our icing sugars at home. Or I can make it into a page divider. This is also dyed, and I've used it to separate my pages and make it into a page divider. So what am I trying to say is that I would like us to look at our wastes and all these are leftovers in a good light. In conclusion, I would like to tell us that these leftovers are not totally bad if we can get to use them the right way. So what I want us to do is, and I'm challenging myself and I'm challenging you also, I want us to get this waste and separate them nicely. How? The waste from my farm activities, I put them in a nice place. The waste from my household, food, leftovers and all, we keep them in a nice way. The waste from plastics that can be recycled, I keep them in a nice way so that these wastes are kept properly. And after they are kept, people that need them or want to use them, me, for instance, I want to use the food waste for my bugs to grow. For instance, the next person in the plastic industry wants to recycle the plastics. The ones that we can't do anything with, we dispose them separately. That way, I'm challenging us to be able to use our waste and separate our waste in a way that it will be useful and it will be easy to use for everyone that is interested in using them. Thank you for listening.
Thank you. That was fantastic. Wow. We have a few questions for you, Dr. Moyinoluwa. Can this be done on a larger scale?
Yes, it can actually. It can. It can be done on a larger scale. So though we need to do some more research, doing it on a larger scale, we think of having a larger bioreactor or larger, yes, it's called bioreactors, fermenters, larger containers. That's a better word. Larger containers that can, where we can use a larger quantity. That way you can use a larger quantity of waste and use your cells. You can put your bugs in them and they would grow it. So the more the container, the more the size of what you get, and the more the bugs you add to it. So, and if we design it to the environment. So for instance, in this research, I didn't have to do anything in a special condition, because these bugs would do very well at room temperature just on the shelf in my laboratory, on the shelf at home. So I don't need any special temperature condition to do this, because of those how those particular bugs are, their own properties.
Fantastic. Someone is asking if you could please just quickly talk about how cellulose is made, the process.
The process. So from trees, right? So from trees, the process is a bit, you cut down the trees, you have to, as you cut down the trees. So either you can get cellulose from either the waste from the trees or from the tree itself. So you cut it down, you remove the bark. After removing the bark, you break them down into chips. Okay, so for instance, like sawdust, and the way we all know the way sawdust is. So you reduce it like sawdust, break it down. After that, you have to go through, there's a process called like pulping, where you like mash and all that. So the idea I want us to have is having paper, like paper mâché. I don't know if we remember how we do that process in school, like paper mâché, where you mash everything. So you have to use like machines after that, or use chemicals. Some people use like water for pulping and all that. After that, you have to clean, separate, because in trees, cellulose is not just alone. Cellulose comes with hemicellulose, it comes with lignin. So if you want to remove just cellulose, you have to separate those things from the cellulose. So that's the process where you have to, lignin is harsh, they call it like a matrix. Is a very strong structure where you have to use a lot of harsh chemicals and maybe temperatures and all. So that you can get out the cellulose, take out the lignin and take out the hemicellulose. So that process is a bit complex, as I'm trying to say. But for bacteria, the cellulose does not come with anything. It is purer form of cellulose. It does not come with hemicellulose or lignin. It's just the bacteria giving you the cellulose and you get it.
Fabulous. Wow. You're making me want to come to your lab and see how you do it. So we have a few comments from the audience. Ume says, wow, this can help with the move against climate change caused by deforestation. And then we have another comment from YouTube from Tina. She says, thank you so much for this presentation, Dr. Moyinoluwa. That was fabulous. Thank you very much. The rest of the questions we'll take at the end. At the end, because of time, we'll take it in the general Q&A. So you will have more. Somebody muted me. Yes, so you'll have more questions at the end, Dr. Moyinoluwa. Oh, thank you so much. So we'll move on to our next speaker. And our next speaker this afternoon is Dr. Rebecca. She's a lecturer and research scientist based at Nnamdi Azikiwe University in Nigeria. She has always been intrigued by the immune system ever since she learned about its ability to affect the way living beings respond and protect themselves from the invasion of harmful organisms during her undergraduate studies. So this interest led her to pursue postgraduate studies in immunology, and she also received a scholarship from the federal government of Nigeria to study in the UK, again, to study her dream course, which is immunology. She's now a senior lecturer and research scientist at the Nnamdi Azikiwe University. So, Dr. Rebecca, it's over to you. put your video on so I can spotlight you, please. Can everyone hear me?
Yes.
Okay. Is Dr. Rebecca here? Oh, I can see her here, but her video, something's wrong. Okay, let's see. If you can hear me, Dr. Rebecca, just say hello, and then I can spotlight you so we can get started.
Yeah, there she is.
Oh, fabulous. There you go. All right. Please unmute yourself. Okay, try again.
Can you hear me now?
Yes. Oh, so go ahead.
Hello, everyone. I'm Rebecca Chukwuanukwu. I am an immunologist. An immunologist is a scientist who researches and studies on the immune system. So you may ask, what then is the immune system? The immune system, simply put, is your own body's defense, its own soldiers, its own guards that protect it against invasion of microorganisms that could cause harm. So this it does using like white blood cells, you've heard about them, the neutrophils, eosinophils, basophils, lymphocytes. All these help in protecting you against microorganisms that could invade against your system and cause disease, cause you to suffer from ailments. So like I said, I'm an immunologist, and we work basically trying to find out how we interact with the organisms that surround us, how we live with the organisms, and the findings from this research is what helps us to find a balance with what we live with all the organisms that we live with in this environment, many of which cannot be seen. So I research on vaccines. Our team has researched on vaccines, and for this COVID vaccine, one of the things we did is that we used vaccine constructs in silico, that is computer generated, to try to provide a construct of the vaccine that could now be used to make vaccines. So these findings have been published in 2020 and 2021 in internationally peer-reviewed journals. So these are vaccine designs that are out there that could be used to make vaccines. So you have different types of vaccines already out there made for use. But the situation now, the problem is that these vaccines, there's fear, there's apathy towards taking these vaccines. Why? The apathy basically is due to lack of knowledge. People are not aware, or people have been programmed or been misinformed into believing that the vaccines could be harmful to us. So one of the reasons I'm having this talk is to try to explain more about the immune system, what it's all about, and how we utilize our immune system, how it is our own immune system that teaches us about how to make vaccines and how these vaccines are used to protect world health, just like we started using them early on. So to the discussion today, I want to ask you, are you aware that we are challenged that we can inhale microorganisms in their thousands every day, some of which are able to cause us disease and are capable of causing us disease and do cause us diseases sometimes? If your answer is yes, you're correct. We actually inhale them in their thousands and even millions. We do not see them, just like you do not see the particles of dust in your system. That doesn't mean they don't exist. But these things are there. They can actually cause us diseases. So you may ask me then, if they are there and we inhale them and they can cause us disease, why do we not fall ill all the time? That is because we have an immune system. Our immune system, like I said, are those soldiers that are there to protect us. They have the capacity, they have the ability to recognize self from non-self, things that are within and those that are not of us. They can recognize them, they can protect us against them, they ignore those that deserve to be ignored, and then destroy those that are capable of causing us diseases known as pathogens. These they must clear from the system if we are going to be healthy. So this is a very elegant system that we have in us that is able to protect us from disease-causing pathogens, disease-causing agents. So that we do not see them does not mean that they are not there. We're able to see them and we have seen them. You can use that magnifying glass to see them, or more elegantly as scientists do, we use the microscope. We use a microscope to look at them, visualize them. The microscopes are able to magnify them to thousands of times, hundreds and thousands of times, so that we see them. Even the parasites, you may not be able to see them, but the microscopes will help you to look at them, see them, and then that's when you can see them, scientists see them, research on them, work on them, and they protect and work on ways to protect us. So ab initio, our immune system, like I said, they have a protective area. So just like a soldier, like this, but I hope you can see this. Just like a soldier will parade to protect us, protect our environment, the integrity of our environment. That is the same way as our immune system has these soldiers. So if for any reason they're able to identify, they zoom into the place, the environment, look around. Some of them, they have this function of seeking for foreign things. Then they zoom in, and they are our supermans, superwomans who come in, fight, really do battle. They actually do battle and destroy those harmful organisms that could come in to try to destroy us, because they have the ability for recognition and then defense. And when they're able to do this, that is when you do not even fall ill. You are not even aware that you have faced a challenge at all from microorganisms that could cause us disease. Another scenario is when these microorganisms actually breach the defenses of your immune system, in which case you fall ill. But have you ever observed that sometimes when you fall ill, you actually get better without drugs? Your immune system has delivered. It has done its job so that it has come in, done the battle, cleared those things from you, and then you are ill, and then you're fine the next minute, and then you continue to move on. Another scenario is when the immune system is totally overwhelmed, in which case you fall ill. So when you take drugs, it may tilt the balance in favor of your immune system so that it wins the battle over these microorganisms that could be trying to establish or have already caused a disease in you, and then you get better. And the other scenario is the bad one, where you suffer debilitating effects of the ailment, and sometimes you can even die. And this happens with a lot of diseases where you suffer from them. But one capability that the immune system has, a very, very important characteristic, that it has what is known as immunologic memory. That is the ability to remember a disease that it has come across before, such that on subsequent challenge, it rises, or that's what I mean by subsequent challenges. If that infection tries to come in to cause you disease again, it rises up much faster because it can remember, it can recognize that it has seen this before, and then protects you against it. So the best case scenario is seen when you have infections and you don't suffer from it again for life. Can you name one or two of them that you've suffered from, but you never suffer from them again? If you mentioned chickenpox, you're correct. If you mentioned smallpox, you're absolutely correct. If you mentioned mumps, you're absolutely correct. These things, once you suffer from them once, your immune system is so trained, and because of the nature of the disease-causing agents, they're exactly the same, so that once your immune system is able to recognize them, it quickly calls on everything it has, and then clears it from your system such that it has no opportunity to cause you disease again. So it is this capability, this nature of the immune system called memory that vaccines utilize. How, you may ask? So what happens in making of the vaccines is that they try to, originally, it started with trying to use chemically modified pathogen disease-causing agents. You can kill them, hit them with use chemicals to make them not harmful to us. However, your immune system is still able to see them as that disease-causing agent and prepares it. So the immune system, the vaccines train your body, trains you to be ready for battle, just in the way that it happens with the natural case. However, it removes the issue of you suffering from the harmful effects. Because the truth of the matter is, when you are challenged with a disease, the different outcomes can happen. You can overcome it, you can fall in with the disease, or you can even die from it. So which means that your immune system could not help you. But what the vaccines will do is that the vaccines will train your immune system without harming you, but will stimulate it. That is what will happen when in situations where you get a vaccine. We all, the childhood vaccines and all of them, you have the vaccine, and then you may experience fever, aches, That is when your immune system is mounting the challenge. It is ready for battle and it is fighting, and it will just be prepared so that on subsequent challenge, it will believe it has seen that disease before. Exactly what is happening with many vaccines that we take, happening also with COVID. It will believe that it has seen the COVID virus, that is SARS-CoV-2 virus, and then is prepared so that you do not suffer from all the effects of this disease, because it is prepared. It has worked. It has been ready for battle. So you cannot compare someone that is prepared. You know there is a war. Your body has trained for it. It is ready for battle, and then it goes into battle. So it blocks everywhere, doesn't allow the system to come in. So, but you may ask, why then do some people suffer differently from diseases? The answer is that we are different. As our faces are different, we also have different abilities. Abilities to, the ability of our immune system to mount a protective, a protection against these pathogens. This could be due to your age. In the elderly and in the very young ones, they have a less robust immune system. But in the prime of our lives, our immune system is at the optimum. That way, people, as you can see, it's easily observed, those that are in the primes of their life do not suffer as much from this COVID as those that are elderly. So these are the things that happen. And then if your immune system is depressed for any reason, say you have an immunosuppressive disease, say pregnancy, because in pregnancy, your immunity is a little depressed in order to allow the baby to survive. These are the situations that will have. Then your diet, then your innate ability, your natural robustness, just like two people can be exposed to something, one will develop something, the other one, the immune system is more robust and blocks it from happening. This is what happens. So in the SARS-CoV-2, what they use is like, there are different types of vaccine constructs. So what is used is, some of them are messenger RNA, so in which you use mRNA, that is the gene coding for the virus, for the SARS-CoV-2 virus. They give it to the people, and they give it to as a vaccine. The immune system recognizes it, uses it to make, recognizes it, and then attacks and then protects you. You also have the ones that use, okay, that's a bit technical anyway, but there are different types. But the important thing is that you give things that are harmless, that do not give you COVID. They do not affect fertility. They do not cause you problems, but they are able to protect massively so that you're ready for battle and are protected from suffering the ill effects of this COVID. So the COVID vaccines are safe. I am a scientist. I am vaccinated. I trust that the vaccines out there, being involved in them as well, that they are out there to protect us. If everyone is protected, if as many people as possible get vaccinated, then the chain is broken. Thank you very much.
Thank you very much, Dr. Rebecca. Thank you so much. We have a couple of questions for you. Let's see. Somebody's asking, what is a vaccine? What is a vaccine? Oh, I think Ethernet has frozen. Okay. All right, I think she's frozen at the moment. Oh, she's back. Dr. Rebecca, can you hear me now? All righty. So we'll save the questions for later. Or if she comes back while I'm introducing the next speaker, I might quickly sneak it in. But let's move on to the next speaker. We will try and put out your questions to her at the end of in the general Q&A. Or Dr. Rebecca, can you hear me? Are you back?
I'm unmuting myself. I can hear you. Yes,
Fab. Okay,
I can hear you.
The first question for you is, what is a vaccine?
So a vaccine, like I was trying to explain, is that it's something that is given which tricks your immune system into believing that it has come in contact with the disease-causing agent, the particular disease agent you're trying to vaccinate against. It does this because it is trying to prepare you for battle. Perchance you come across the real disease-causing agents that will now cause you a lot of harm. So it does this without you suffering from the ill effects of the disease itself.
Okay, so then how does the immune system know which bacteria are bad and which ones are good?
Good question. Thank you very much for that. The disease-causing agents were found by scientists that they have something known as the pathogen-associated molecular patterns. These pathogen-associated molecular patterns are associated only with those that can cause disease. So you have many microorganisms in the body. You have those ones that can be on your body but are not harmful to you. You have those that break down your food that you eat, and so on and so forth. Those ones do not have these pathogen-associated molecular patterns. However, those that have the pathogen-associated molecular patterns, your immune system has been trained to recognize them with things known as pathogen recognition receptors. So anyone that is a pathogen, they will recognize them and alert the immune cells in your body to run into battle. So all of them migrate to the site that thing is trying to come into your system and battle it.
Amazing. Amazing. The body is just amazing. Fantastic.
Wonderful. Fantastic.
Thank you so much. Because of time, we'll move on to the next speaker, and then we'll take more questions for you in the general Q&A. Thank you very much. Thank you very much. A few comments for you, Dr. Rebecca. Great presentation. That was very interesting. So thank you. Thank you once again. All right. So our next speaker. I hope everybody is still paying attention and are absorbing all this fantastic information that we're receiving today. Good. All right. So our next speaker, Professor Omonkhua. She obtained her bachelor's and master's degrees in biochemistry from the University of Benin. And then she obtained her PhD in toxicology in 2010. Her PhD thesis was nominated for the NUC Doctoral Award in 2012. She's an active researcher, and as she said earlier, she's a teacher. She loves teaching. Her major focus is on local medicinal plants used to treat diabetes, malaria, and reproductive disorders. So, Professor Akhere, over to you.
Thank you very much. Do you know that diabetes is one of the oldest diseases ever described? The first mention of this disease was over 4,000 years ago. However, it was not until the early 1900s that the cause of this disease was described. Scientists now know that diabetes is caused when the person cannot use or cannot produce or use a substance called insulin. Diabetes is a chronic disease. This means that it lasts throughout the life of the person. Do you know that 5 out of every 10 medicines is derived from medicinal plants? Well, can medicinal plants be used to treat diabetes? Well, I think before I answer those questions, let me first of all explain what happens in a person who has diabetes. When you eat a meal that is rich in carbohydrates, let's say rice, bread, cake, I know we love that, your body breaks it down to produce sugars. And this sugar is absorbed into the blood. For a short period of time, the blood sugar level will increase. This is normal, and it is called postprandial hyperglycemia. This increase in blood sugar level is noticed by a small organ called the pancreas. And this pancreas produces insulin, and insulin helps your body to use the sugar. So how does it do this? How does it help your body to use sugar? Insulin opens the door of tiny parts of your body called the cell and allows the sugar to move from the blood into the cell. Inside the cell, the sugar is stored so that you can use it later on, or it is used directly to produce energy. And this is the energy you use for all your favorite activities like walking, running, swimming, cooking, and even talking like I'm doing now. And so this reduces the entry of sugar from your blood into the cell reduces your blood sugar level and you're fine. For persons who have diabetes, either they can't produce insulin at all, and in this case, we call it type 1 diabetes, or they cannot use properly the insulin that they produce, and we call this type 2 diabetes. What happens with these persons is that when they eat a meal that is rich in carbohydrates, their blood sugar level does not come back to normal as quickly as it should. Another problem is that the sugar remains in their blood and it doesn't enter into the cell where it's used to produce energy. And so they don't have much energy, they can be weak. Now, diabetes can be treated in many ways. If you are chubby, your doctor may ask you to lose weight. Some doctors may also prescribe exercise and a diet change. This is important, especially when you introduce diets where the breakdown of this diet does not produce too much sugar, and so it can help you regulate your blood sugar level. Some doctors can also prescribe drugs that can lower your blood sugar level. If all of these do not work, or if the person does not produce insulin at all, the doctor will prescribe a daily injection of insulin to help the person stay well and alive. Now, in spite of all these wonderful drugs that are available, people who have diabetes produce harmful substances that later on cause damage to several tissues in their body, like the kidney, the heart, the brain, where it can actually cause strokes sometimes. And then you have the blood vessels, where diabetes and its complications can reduce the blood vessels and increase blood pressure. And then for some people who do not manage their blood sugar levels, it can lead to sores and ulcers in their foot. And if it is not managed properly, it can lead to the amputation of the foot. Another thing that worries scientists is that many more people are developing diabetes. And so they're curious and they want to find out ways, better ways of preventing and treating this disease. Like detectives, one of our clues comes from the fact that our forefathers have used medicinal plants to treat this disease for a very long period of time. And some of those medicinal plants include a very common bitter leaf. And then we have scent leaf, you know the one, smells really nice. And then we have soursop leaves. In my lab, we have looked at 4 of these medicinal plants that have been used by our forefathers to treat diabetes. And we have found out that 2 of them have very good effects in lowering blood sugar level. And they don't do this in just one way, they do it in several ways. The first way is that they reduce the rate at which carbohydrate-containing foods are digested, and so they slow down digestion. Number 2 is that they reduce the rate at which these sugars that are produced enter into the blood. So that sharp increase that you see in blood sugar level does not happen. Number 3 is that some of these plants actually stimulate the production of insulin. And of course, when insulin is present, it can do its job. And then finally, some of these plants help the body to use insulin properly. And so remember that insulin opens that door of the cell. These plants actually help them to open the door better so that the sugar can enter into the cell and the cell can use it to produce energy. Another interesting thing is that apart from lowering this blood sugar level, these medicinal plants reduce those harmful substances that can lead to those long-term complications to the eye, to the kidney, and other things like that. And so now that we know that these plants do this, we now want to find out what is inside this plant that is causing them to have this effect that are positive in treating diabetes. And this is a tricky part of our experiments for 2 reasons. Number 1, medicinal plants contain a lot of chemicals, thousands of chemicals. So how do we now know which one is responsible for its ability to treat diabetes? So what we do is that we continue to subdivide the plant. Every time we subdivide, we check to see whether it still has the ability to treat diabetes. And then we'll continue this until we reach the chemicals or group of chemicals that are responsible for this effect. We call this process bio-guided fractionation. The other tricky part is that sometimes when you do these subdivisions, you may lose some beneficial substances, whereas the substance you're looking for specifically becomes more pronounced. It is possible that some of those beneficial substances you have lost would have been good in helping the plant fight diabetes. But if your aim is to get to the chemicals that are responsible for this ability to treat diabetes, this step is inevitable. You have to proceed with it. Apart from finding out what is in these chemicals that is causing them to have this effect, we also want to know how they do this. We call this the mechanism of action. So when we have been able to answer these two questions, the questions are, number one, what is in this chemical, this plant that is causing it to have this ability to treat diabetes? And number two, how does it do it? We can now move on to other studies where we look at the effect in human diabetes. We want to see whether this can have the same effect on humans. So we'll go back to one of the questions I asked at the beginning. Can medicinal plants be used to treat diabetes? The answer is yes. Work from our laboratory, and indeed several laboratories all over the world, show that this is true. And not only can they reduce blood sugar level, they can also reduce the harmful substances that lead to long-term complications of diabetes, like eye damage, foot damage, and the rest. And who knows, we may be able to add to the growing number of drugs that come from the rich world of medicinal plants. Thank you.
Thank you. Wow, that was so interesting. Very, very interesting. We have a comment from Ume saying, gosh, very interesting. Africa is so blessed. We are indeed. We have a few questions for you. Can diabetic patients consume these plants in its raw forms with the hope of managing disease? Are we at that stage to know this?
Okay, thank you so much for that question. I get that a lot all the time. Now, when a person is already diabetic, you really have to see your doctor and get it sorted out. Most of these drugs, most of these plants are currently in the stage of being processed to see what chemical is in them. Remember that I said that many medicinal plants have thousands of chemicals. And so if you're eating this the medicinal plants as food, it is fine. But if you're using it to treat acute diabetes, you are not using the concentrated chemicals. That is the main chemical that is responsible for its anti-diabetic effect. So it may take a long period of time for it to have an effect, and you don't have that kind of luxury. You should see a doctor and attend to the acute nature of the disease. What most plants do, especially when you take vegetables and fruits, is to build your immune system and protect you against diseases. When we're able to find that exact chemical or group of chemicals that is responsible for this, yes, we could say we can use that. But at this stage, we can't say for sure whether by eating just the food you can get this effect. But note that most of these plants are plants that we already eat, so there's nothing wrong with you eating them for food.
Okay. Yeah, so how close do you think you are in finding what this chemical is these plants are? How close?
Okay. For one of them, pretty close. Just about 2 months ago, I sent the chemical, the isolated compounds out for proper identification. And they are in the process of identifying, actually sent 13 of the compounds we isolated. So once we've identified the structure of the compound, we'll do further studies to see how they relate with substances in the body that tell us that they are anti-diabetic. We can use computer simulations for that. I think I'll explain that. Once you get the structure of the compound, you convert that structure into a 3-dimensional form and then use it to check whether it affects a specific anti-diabetic or diabetes-producing structure in the body. If it protects against it, then you have your answer. that this is a positive compound against diabetes. And we can now explore that further. So pretty close, I would say.
Oh, it's so exciting. It's really exciting. Yeah, great work. We have a comment from Rose. Thank you, Professor Omonkhua, for an excellent presentation. And one of your mentees has also left a message. So they are here listening. We have more questions for you, but again, we will leave it at the end to the general Q&A. Comment from Tina, very interesting. Yes, it is, Tina, very, very. It's exciting. There's a lot of things that could happen if this breaks through. It would be amazing. Thank you so much, Professor. So we'll see you again later.
Thank you.
All righty. Okay, so our next speaker is Ifeoluwa Peace Oladapo. She is a graduate of the University of Ibadan, where she studied her undergraduate degree in zoology and also completed her master's in parasitology. She's been involved in a number of health-related research work, and she's currently about to enroll for her doctorate degree. Ifeoluwa, over to you. Where is your? Please put your video on so I can see you and spotlight you. There you go. Fantastic.
Yeah. So good afternoon, everybody. My name is Ifeoluwa Oladapo, a trained parasitologist, well, someone who studies parasites. So for this program, I'll try as much as possible not to use scientific terms and break it down as much as possible so that the people that are not science related can get what I'm saying. So today I'm going to talk about the scientific facts about toxoplasmosis, Toxoplasma gondii. So we're all going on a fantastic ride to discover a guy who just wants to keep collecting from you without giving you anything in return. This guy just wants to exploit you and feed on you, and sometimes leaves you broken and sick. This guy is called a parasite. So in a nutshell, a parasite is somebody or something that feeds on you at your own expense, that exploits you, takes from you, or even leave you sick or broken. So an example of this guy is Toxoplasma gondii, what I'll be talking about today. So it simply means Toxoplasma gondii is a parasite who lives or feeds on you at your own expense without you giving it anything in return. So for the sake of this program, we're going to address Toxoplasma gondii as Mr. T, so that people that are not science oriented would quickly get or grasp what I'm trying to talk about. So Mr. T is a very tiny organism that cannot be seen with the bare eyes. What do I mean? As little as an ant is, we can all see an ant with our bare eyes. But Mr. T, which is Toxoplasma gondii, is so tiny that it cannot be seen with the bare eyes, and one needs an instrument to be able to enlarge it, for it to be enlarged, or for it to be magnified, to be seen. And this instrument is called a microscope. So it means a microscope is an instrument that lets you view things that you can ordinarily not see with your bare eyes. So where does Mr. T, Toxoplasma gondii, don't forget that Toxoplasma gondii is being referred to as Mr. T because of this program. So where does Mr. T? He lives in meat, like beef, pork. Mr. T resides basically in dirty places. Mr. T resides in the soil. Mr. T resides in cats and on fruits and vegetables. So who can get Mr. T? Anyone can get Mr. T, except maybe my bicycle. That sounds funny, but yes, anyone can get Mr. T. So it simply means that anyone who has blood flowing in their veins can get Mr. T, be it animals, be it humans, adults, babies, irrespective of age, irrespective of your gender, whoever you are, you can get Mr. T. So as much as possible, because in our part of the world, in Nigeria, not so many people are exposed to Mr. T. People don't even know who Mr. T is. People have not heard about Toxoplasma gondii because it is neglected in this part of the world. This in the US, when you are gravid, it means when you are pregnant, it is compulsory for you to undergo a Toxoplasma antigen test. But here in Nigeria, people don't even know what the parasite is. Let's talk of you going to do a test to know whether you have it or not. So it is so neglected that it's poorly spoken about. People don't know it. People don't even know what it looks like. People don't know the do's and the don'ts that are attributed to Mr. T, and it is not communicated to the general public. So because of this program, how will one know that, okay, I have Mr. T, or Mr. T has visited me? Or how will I know that I have come down with Mr. T? Normally, if one has malaria, you will know that, yes, this is malaria to an extent. But when you have Mr. T, how will you know that Mr. T has actually visited you? It is so interesting that Mr. T can live with you. You can have it in your body system. It can be with you for a long time, and you wouldn't know. This is because there will be basically no major changes in your body. There will be no sign. There will be no, you might even feel the normal way that you feel. So that's how bad Mr. T is. Mr. T can reside in you if your immune system, like one of the speakers spoke about the immune system earlier on. So if your immune system is very strong, you can have Mr. T in you for a long time. But people that their immune system has been compromised can come down with some signs, which includes fever, maybe headache, body ache, and stomach ache, as the case may be. And there are some behaviors that can expose one to Toxoplasma, which is Mr. T. For example, like I said earlier, where Mr. T resides and lives. For example, like I said earlier, I said Mr. T lives in the meat. Okay, as ladies or women or even men and guys, just imagine that you are in the kitchen trying to make meat, and you want to be sure that this meat is really cooked, like it's cooked, well cooked. And maybe 5 minutes or 10 minutes after you are boiling the meat, you just go there and take a bite. It is possible that Mr. T is in that particular meat that you just took a bite of, and peradventure you just introduced Mr. T into your body system. Also, for women or anybody that goes to the market to purchase fruits and vegetables, you can buy fruit and want to be sure that this particular fruit is maybe ripe or sweet or something. For example, apple. And we just pick up the apple and we're like, oh, let me just taste it, let me have a bite to know if I want to buy more or not. As you just took a bite without rinsing, without properly washing. It's possible that Mr. T, which is Toxoplasma, is on that particular fruit, the apple. And you consuming it like that, it means that you're consuming the apple together with Mr. T, and you're predisposing yourself to be infected by Mr. T. Like I said Mr. T lives on fruits and vegetables. Also, for people that have cats, I like cats. Cat is the definitive host of Mr. T. What do I mean? It means that the life cycle of Toxoplasma gondii, which is Mr. T, is completed in a cat, and it comes out. The infective stage comes out together with the cat's poop, like the cat's fecal matter. So if you are this type of person that has cats in your house and you want to change the cat litters, like the poop of the cat, with your bare hands, it's possible that the infective stage of Mr. T, which has been passed out by your cat, together with the cat's fecal matter, it's possible that as you are trying to clear up, you have also picked up the infective stages in your hands. And peradventure, maybe as you were walking, there's something beside you that you are just snacking on at the moment. I'm walking, I'll snack on this. It's possible that as you want to snack on whatever it is you're snacking on, you're also introducing Mr. T into your body system. So, for example, peradventure, there are different ways to prevent Mr. T. Like I said earlier, Mr. T resides basically in dirty places. So one of the things to do in order to prevent Mr. T is regular hand washing. And thank God for the era, well, yes, for COVID-19. Now it's the normal thing, it's part of our normal duties to wash our hands, even though that's the normal thing we're supposed to do normally. But now, thank God for COVID-19, I think it has also helped to increase the way we wash our hands, sanitize our hands and all. So also, we should avoid undercooked meat. If you want to cook your meat, if you want to boil meat, make sure the meat is well and properly boiled. Don't consume half or undercooked meat. When you want to change your cat's litters or poops, make sure you wear gloves or even if you wear gloves, make sure you wash your hands like almost immediately after whatever you are doing. Then also for the people that work like farmers that comes in contact with soil regularly. One place where the infective stage of the Toxoplasma gondii, which is the part of the parasite that infects people, lives in the soil. So people that are farmers or people that does things that are related to soil, if you can't wear gloves, because some people are not comfortable wearing gloves, because some people will tell you that I can't wear gloves because it slows me down. If you know you can't wear gloves, as soon as you are done, make sure you wash your hand. Like my research team and I, we work on Toxoplasma gondii basically, and it is interesting to know that a lot of people out there don't even know what Toxoplasma gondii is. A lot of people have never heard about it before, because one of our researches that we did, we put out a questionnaire to know people's belief, their attitudes towards Toxoplasma gondii. But it's very sad and disheartening that in a population of, okay, for example, let's say 100%, just 10% knows about Toxoplasma gondii. Not so many people knows about this particular parasite. A research was done by my team and I in Ibadan to know the prevalence of Toxoplasma gondii amongst residents. When I say prevalence, I'm talking about the percentage of the people that are positive to Toxoplasma gondii. And we reported over 60% prevalence, which means, yes, this parasite is out there, but people don't know about it. Do you get? So I think one of the reasons why I'm coming out today in this program is to sensitize us about Toxoplasma gondii, to make us know that, yes, there's a parasite called Toxoplasma gondii, which is poorly spoken about, the do's and the don'ts, what you can do to prevent yourself from it, and all. And I believe that with this little talk, I believe that we now know what Toxoplasma gondii is. I believe that we now know things can prevent ourselves from coming in contact with Toxoplasma gondii. And I hope that I go out as well. Okay. Thank you very much.
Thank you very much, Ifeoluwa. So you mentioned that you and your team have done some research in the lab on Mr. T, as you call it. So what research has been done on the effect of Mr. T? Or what other research have you and your team been conducted in the lab on Mr. T? Please unmute yourself. Please unmute yourself. There we go.
Yeah. So, for example, the new school of thought that Mr. T, Toxoplasma gondii, is related or has a link to schizophrenia. This is schizophrenia. So my team and I, we actually went to UCH, the psychiatric ward in UCH. We collected blood samples where we checked out for the parasites, Mr. T, in their blood samples, to know if it is a contributing factor to their current state of health, so which will also inform how they will be treated and all. So we checked out for the dopamine level, the serotonin level in them, and it was interesting to see that we did the IgG and IgM. When I say IgG is an antibody to tell us that currently this particular person has this parasite. And when I say IgG, it means that this person has at one time been exposed to this parasite. And their IgM level, the prevalence was 64.8%, which means that in those categories of participants, it's possible that it is a contributing factor to their current state of health. But a lot of people don't know this. Also, we did another one in Ibadan with pregnant women, because it has also been said to be linked with to congenital issues, and it was also prevalent. It was on the prevalence was on the eye side, and people still don't know about this particular parasite.
Wow, that's very interesting research going on. And are you still working on that?
Yes, at the moment.
Okay, fabulous.
Across different geopolitical zones.
Okay, oh, that's great. So just one question. Somebody asked, what is a parasite? So if you can just answer that within just 30 seconds for me, because of time. What is a parasite?
What a parasite is Okay, I want to use scientific things. A parasite is something that lives on you at your own expense. Something that feeds on you at your own expense. Something that just wants to take from you at your own expense. So what does this mean? It means that you are in a relationship with something, but the thing is not giving you anything, but you are the one giving the thing something. And as you are giving the thing something, the thing is either leaving you broken or even kills you sometimes. Do you get?
Yeah.
Does that answer the question?
Yes, I think so. Thank you very much. Just to go back about your study on the potential effects of Mr. T on schizophrenia, you are just studying the potential effects, isn't it? It's not that it causes it. We don't know that yet. You're just studying the effects. That's right, isn't it?
No. Yeah, we're just studying the effects because there's a link between it. So we're trying to ascertain if there's really a link between it or not.
Thank you very much. All righty. Thank you so much. Let's move on to the next speaker. Any more questions for you? We'll do that at the end. Okay, so our next speaker, last but not least, is Dr. Oluwakemi Oyelowo. She's a lecturer at the University of Lagos, and she has a PhD in physiology, and she's also been lecturing for over a decade. The aim of her research is discovering ways to prevent disease and improve health, and especially in terms of reproduction. Her work focuses on the roles that diet, endocrine, and environmental pollutants play in the physiology of an organism, and how to better such effects, especially along the reproductive axis. Dr. Oyelowo, over to you. Let's see. You got your video on? Is Dr. Oyelowo here? Oh, there you go. All righty. Okay, so just unmute yourself. Can you hear me, Dr. Oyelowo?
Yes, I can hear you.
All right, go ahead.
Thank you. Eating safe and healthy is important for health and wellness. So it is important that we are aware and updated on food items that are taken regularly. Do you know that seeds are edible? Do you even know that they are edible seeds? If you do, please take a piece of paper and write out 5 seeds that you know that are edible. Did you write pumpkin seeds, pomegranate, sesame, canola seed, African oil bean seed, oil palm seed, granite, melon, maize, and so on, other examples? If you did, well done. Seeds are no longer thrown away as wastes because there is renewed interest in them because they have been found to have promising effects on human well-being. Seeds also contain vitamins and minerals that are important for the body. And another thing to note concerning seeds is the age and maturity of the seed. Are the seeds from ripe fruits or unripe fruits? Now, the seed of interest for today's talk is pawpaw, and the botanical name is Tariqa Papaya. Tariqa Papaya has heart-protecting effects. It protects the skin. It also controls sugar level, and it is known to fight diseases in the body. Now, Tariqa Papaya is used in some cultures in Asia and Africa as a replacement for black seed. And it is also used in some cultures in Nigeria and some other places as a replacement for locust beans. So it is safe to say that Tariqa Papaya is eaten in the adult population. What is not known, however, is the effect of Tariqa Papaya on pregnancy, especially if the seeds are from ripe fruits. Now, this is how unripe fruits look like. the unripe fruit and the seed. They look green. And this is how the ripe fruits look like. Now, in our laboratory, we got pawpaw seeds and we processed them and we gave them to albino rats, pregnant albino rats. Now, albino rats are used in the laboratory to carry out, experiments are carried using them because they mimic what will happen in the human. So we gave these pawpaw seeds to pregnant albino rats, and we gave them during the first trimester of pregnancy, the second trimester of pregnancy, or the first stage of pregnancy and the second stage of pregnancy, if you like. And we also gave some throughout pregnancy. Now, what we discovered was that in those that consumed the seeds of pawpaw from the ripe fruits during pregnancy, all through pregnancy, had their pups dead. Pups are baby rats. That's the name given to baby rats. They had all their pups dead. While those that were administered the seeds of the pawpaw seeds from ripe fruits had pups. Those that were given during the first stage of pregnancy and the second stage of pregnancy had pups. Now, an interesting result from this study is that we discovered that these pups, these baby rats, were very big. That is, those that were given during the first trimester of pregnancy and the second trimester of pregnancy. They had their pups, they were big when they were born. And another interesting thing is that in almost all of them, there were more males than females. So we decided to find out what would be the effect of this pawpaw that their mothers took during pregnancy on their bodily appearance and also up until puberty. Puberty is when the childbearing organs mature. So we decided to study them over time. And now, usually when rats are born, they are usually born blind. Their babies are blind, and then over time as they grow, their eyes open, their ears open, there is hair on their body known as fur, so they stop looking pinkish like this. Now, what we discovered was that in these pups whose mothers took the ripe seeds of pawpaw, we discovered that their eyes did not open on time. There was a delay in eye opening. Their ears did not open on time. The fur, the hair that was meant to be on the body also did not come out on time. Their teeth, the incisor, came out earlier than expected. And of course, when they reached puberty, there was also a delay in the age of puberty. They didn't get to puberty on time. So by implication, when these male pups are, get to the age of childbearing, they will have issues. There were also alterations in their hormones. Hormones are found in the body. They help the cells and organs to work. Their reproductive hormones were altered. And the stress hormone in the body was we also discovered that the level of the stress hormone, when it was measured, was very high, indicating that these seeds had some alterations, in them. Now, these experiments will also be, please take note that these experiments will also, there will be further experiments, there will be further laboratory experiments to actually subdivide the components of these pawpaw seeds from ripe fruits to know exactly what is causing these effects in the offspring, in the babies. And subsequently, and then further in future, we hope that there will be clinical trials that will scientifically show if it is safe to consume these seeds from ripe fruits in pregnancy. Please remember that it is important to eat healthy and safe. And in pregnancy, consumption or caution should be taken when consuming seeds even when they are from ripe fruits. Thank you very much for listening.
Thank you so much, Dr. Oyelowo. So we have a question for you. What is the effect of consuming seeds from unripe fruits in pregnancy?
All right.
What is the effect on pregnancy? Yeah.
Yes. Research has shown that unripe fruits, or they are usually called greens, you know, they are not ripe. When they are not ripe, they contain different compounds that are toxic for the unborn baby. And this makes the uterus where the baby stays to be, you know, to be tight. I mean, in a layman's word, to be tight. And so it shrinks the baby and anything can happen in those kind of conditions. I mean, in laboratory studies, it shrunk the baby animals that were administered, whose mothers took these unripe seeds. So they are not advised to be taken, so those experiments show that unripe seeds could be harmful. That was what the laboratory studies reported.
Thank you very much for that. Another question is, what are clinical trials?
Okay, clinical trials are trials that are carried out on humans. Now, there will be there are usually the stages of research is such that the experiments are first carried out on laboratory animals. There are so many of them: rats, rabbits, and so on, monkeys, and so on, depending on the experiment, dogs. And then after different stages, it will now be carried on human beings. There will be volunteers and all that who will be able to ascertain if it is if it is important, or if what will be the effect on humans.
Thank you for that. That was a very interesting talk. Have comments in the chat saying thank you, that was interesting. Someone has said, I will look at seeds with a different eye now from the information you have given us in your presentation. Thank you very much for that. Okay, so we have come to the end of our speakers that we've lined up for you today. So we are going to go into the general question and answer session. So I would like all our speakers to please turn their videos on so we can have the general Q&A. If you are in the audience here live on Zoom and you would like to ask a question or pose a question or to our speakers, please kindly raise your hand and then I can call you and we can take it from there. All right, so let me just ask our speakers to unmute. And then we can spotlight them. Just give me 1 second. While I'm doing this, please think of questions to ask our speakers. The research is fascinating. So let's grill them. You might ask a question they haven't thought about now. So that can be another research question for them to look into in their field. So please do ask your questions. Okay, so I saw someone put their hand up. Okay, that's fine. Right. Type your question in the chat, please, and then we can call you out, ask you to put your hand up, and then we can ask you to unmute if we're going to take your question. Right, let's go back to the ones we had earlier on. Dr. Moyinoluwa, they're really interested in your cellulose. Please, can you take the process of making cellulose again?
Okay, so the process of making cellulose from bacteria, right?
Yes.
Okay. First thing first, it's a bacteria that is making the cellulose. So you must get the bacteria first, meaning you must get the isolate. We call it, from microbiology, we call it isolation of the bacteria. So now, how do I get the bacteria? generally cellulose-making bacteria are gotten from, isolated from sugary stuffs or waste. So when I say sugary material, so for instance, my rotten fruits has cellulose in it. A cellulose-producing bacteria. Flower nectar also has nectar from flower. Also, I can get it from palm wine that we assume is like, I call it over fermented palm wine that you just left on the table, nobody drank it, and it has turned to trash. So we can get the bacteria making cellulose from there. So after that, I get my material. So in microbiology, we call it the medium. That's what the bacteria will feed on, or what it will grow on, what it will eat to grow and now produce. So for a bacteria to make cellulose, it needs something that it will eat. And the carbon source or the sugar, it is very important. So that means that if I am getting my watermelon rind back, my yam peel, those things are rich in the sugars that the bacteria needs to make cellulose. So all I need to do is get those materials. And for instance, earlier research I did on fruit peels, I didn't have to do anything special to the fruit peel. Even the fruit pulp from a juice industry, once I worked with a pineapple pulp from a pineapple making industry, I didn't need to do anything to it. All I needed to do is just dry it and grind it and put it in liquid form and put my bacteria on it. Yes. Or in it, and it grows. So the cellulose grows, we say it is, the bacteria produces it, I call it produces, the process of synthesis, in the air-liquid interface of the medium. So you have a plastic container and something like this tissue paper. So the top of this tissue paper here, so this is the medium, the cellulose will grow on the surface. So let's say this is where the liquid stops. It starts to grow. When the bacteria produces it, it starts to form. So it's in the circular surface. It starts to grow in circular form. If it's in a rectangular container, it will grow in a rectangular form. It just keeps coming up on the surface. From like the third day of production, it starts coming. So it comes in layers. So it's like one layer is formed, the next layer forms, it goes down. So this is a perfect, let me see. So I don't know how well we can see this picture. It looks like it wants to rain here. So it's like
put it very close to the camera of your laptop.
So I don't know if we can see this. So we see in that medium, there's something like a material there's something that is like whitish, or maybe just taking up the color of the medium, and it is forming like layers down. So that's how it comes in the medium. So it's like the more you leave it, the more it grows. And it keeps coming down over the surface. And after the bacteria gives us that, I just use anything, a tweezer, something that is sterile clean, to pick it out from the medium, just out like that, and you wash it until you are sure that it's free of anything that is, because actually in some places this is produced. I don't know if I've heard of kombucha. Kombucha is a drink. Some people take it. It's in most Middle East, Indonesia, Philippines, and all those places. So they take that kombucha. Some people can eat it directly. If you are making it with maybe green tea or something, you can eat it directly. So it does not have anything, since if you know what you are using to do it. Okay. So it's not from the lab. You don't consume something from the lab. If it is homemade, you can.
Thank you very much for that, for explaining that again. The next question we have is for Dr. Rebecca. And the question is, I had it now and it's disappeared on me. Yes, it's a question from YouTube from Tina. Why do people still get COVID after taking the vaccine? Unmute for us, please.
Thank you. Very, very interesting question, Tina. yes, like I was explaining during my talk, there's a lot of variety of things that could affect our immune system, including how our innate ability, the age of the individual who is taking the vaccine. So your immune system, everybody's strength of the immune system is not the same. Some people, their immune system is so strong that they rise up to the challenge and are able to be optimal to protect. Some others, their immune system is not as strong, and so they fall for it. And then if it's a disease or something, if there's a mutation, remember everybody was talking about a mutation, this variant or that variant, that is happening with COVID. A mutation, the mutation is in there. And when the mutation comes in, the immune system is not able to immediately or quickly recognize it as that same thing it has seen before, which means that there's really like the first area of the person suffering from it, and the immune system now rises up to the challenge. But what is clear is that anybody that is vaccinated has a higher chance of having less adverse effects from the disease. So it is recognizing it partially, but not totally. So mutation is a huge issue, which is why people, like there was a question that was there, why do people suffer from malaria and suffer from it all over and all over again? That is because the plasmodium is highly mutative. It changes so much that your immune system is like thinking it's meeting something, a new thing, which means people keep on suffering from it. Once you're used to that one, another one comes in. Common cold virus also does the same thing, keeps on mutating. And so this SARS-CoV-2 is like, it's like the common, like a virus, a cold virus. So that's it, mutation. Another question, that I saw, someone was asking something very important, which people are worried about, that drugs and vaccines take a lot of time to go out there. So how come this COVID vaccine is out there? Are we sure it's safe and so on? Yes, very important question.
So just 1 second, okay. Dr. Rebecca. Yeah, let's pause. Right. Yeah, just hold on for that. Right. Thank you. All right. So Dr. Akhere, I remember there was a question earlier on that if you have diabetes, how can you control it with diet?
Okay. So one of the major things with diabetes is that there are two types, type 1 and type 2. For type 1, the person does not produce insulin at all. And so that type requires a daily injection of insulin. For type 2, the person produces insulin but doesn't use it properly. And so some people control it by losing weight if they are big. Studies have shown that when people lose weight, their bodies respond better to insulin. Also, people can go on can take a dietary route. So one of the major problems is that your body is not able to handle sugars. So go for a diet where too much sugar is not produced. And then a lot of people go to use a lot of fruits and vegetables. Apart from the studies that we have done, where we have seen that these fruits and vegetables can protect against long-term complications of diabetes. We all know that fruits and vegetables contains minerals, properties that build your immune system, and so helps you protect you against this disease. So yes, you can use diet, but not for type 1 diabetes, please, and not for acute type 2 diabetes. A good diet usually can even protect you against developing diabetes on the long run. And it is something you need to stick with. People diet and they do the diet today and tomorrow they jump off it and things like that. So you have to stick with it and do the right thing, eat properly, and then you get the good results that you want. I don't know if that answers that question.
Yes, oh yeah, that helps. Thank you very much. Yeah. There's another question for you, and it is why? Hold on, the chat is going up and up, and I had the question just now. So it's about your research. Let's just go down. About extraction. Just give me 1 second. It's from P-Man. Has your research attained a synthetic extraction of the required constituents to treat diabetes?
Okay. For one of the plants, we are almost there. Like I mentioned earlier, just last month, yes, we were able to isolate. In fact, about 2 months ago, we were able to isolate about 5 compounds, and some of them we had to co-isolate. Sometimes compounds don't separate properly, they come out together. And so we have about another 5 of that. Now we have to identify those compounds. And unfortunately, we don't have all the facilities here. So we had to send them for, identify the substance. So once you identify it, you can now have a structure of that compound. And that compound can be, computer programs can be used to build a 3-dimensional structure. It is at that stage you can do a synthesis. And in research, it's not just one person's job. If I'm going to get to that level, I need to work with chemists. Remember, I'm a biochemist. So I now need to work. So that's where the collaborative work comes in. Mm-hmm. So I need to work with a chemist to synthesize those compounds. Thank you.
Excellent. Thank you very much for that. Dr. Moyinoluwa, someone is asking how expensive is the process, your process for synthesizing cellulose? How expensive is the process?
The only part that may be expensive, it's the part where we need to design a bioreactor. So I've come to, because my research has pushed me towards alternatives, and we're in Nigeria, we always try to look for the alternative to everything because we can't afford most of the things. So for instance, there's something that there's work that needs to be done on. When we're talking of growing bacteria, you talk of their carbon sources and their nitrogen sources and other nutrients. So if we can get a carbon source like our wastes that are rich in carbon sources, and also get other materials that are rich in other mineral sources, and we have a lot of wastes that are rich in all these things. So if we can use those wastes and expand it, then it can be affordable. Then the part that may be expensive is designing a bioreactor. So our first problem is, when we design a bioreactor, we need to talk on the air, aeration, temperature and all those things. So if our process is less is aerobic, we don't need to look for something that is anaerobic, that needs carbon dioxide, and we have to cover it up. If we can do it in an open space and just get a more sterile condition, we can make it cheaper if we can develop locally made or locally available substitutes. But research has to go, work has to go into that. A lot of work has to go into that. Getting locally available materials is doable, and it will make the cost very, very efficient. Because if bacteria are lying fallow, they are not doing anything. The wastes are there. They are constituting nuisance. We have everything, but how to now bring everything together and make it very cheap? More work needs to be done on that.
Fabulous. As someone has mentioned in the comment section, that in science in Nigeria, you have to be very creative. They say necessity is the mother of invention. When you have to find cheap alternatives. So yes, okay, thank you for that. The next question. And before I ask the next question, please note we are not here to provide any form of medical advice. Thank you very much, Dr. Oyelowo. So the person wants to know if the fruits themselves are safe for pregnant women. Let me just read it exactly as they say. I want to know if the fruit itself is safe for pregnant women. We understand that ripe pawpaw seeds may not be good for pregnant women, but is it safe for pregnant women to eat the fruit itself? Unmute yourself, please. Did you hear the question?
Yes, I did.
Okay.
Yes, okay, so like I said, the studies we carried out are on laboratory animals. And for instance, in this talk that I made, it was interesting to note that even while the seeds were ripe, they still cause alterations in the babies or pups that were born. So in pregnant women, and then like I said, for this study, we'll still have to go for that on clinical trials. Now, for human beings, pregnant women, I would advise that they see their dietitians, because sometimes even some fruits that we take normally are classified as unsafe. So it would be better for pregnant women to see their qualified health professionals concerning what fruits they should take during pregnancy.
Okay, thank you very much.
May I come in there?
Yes, go ahead.
Dr. Oyelowo, please, could you just talk us through, the differences between laboratory tests on animals before we get to human clinical trials? I think that might be beneficial for some of the audience because it's one of the steps, isn't it, towards drug discovery?
Yes, sure. First, the whatever study one is carrying out will first be carried out on small animals known as rodents like rats or rabbits, and then it will go on to higher animals like primates, those that are closer to humans, before we now go on to clinical trials, to be able to ascertain what the results will be in humans. And then these laboratory studies are also in different stages. First, there is the, what do you call it now? How do I explain it? There's a raw plant, for instance, like the pawpaw seeds, like the pawpaw seeds, that's the raw seed. And then there will also be other studies that will break it down, so divide these seeds to know exactly what they contain that are causing this and this and this effect that one is looking at. So before, they will now go on to, so that will also take a while, and then before you now go on to clinical trials. I hope that is clear enough.
Thank you very much. Thank you. Thank you very much for that. Right. Okay. I know we had some questions earlier. I'm not sure we've covered them. Just give me 1 minute. But if anyone has any more questions, please do pop it in the chat quickly before we go. Okay. So a question for Dr. Rebecca from Mona Lisa. Says, thank you, Ma, for a wonderful session. But speaking of vaccines, especially the COVID vaccine, I learned that vaccines and drugs in general take about 2 to 3 years to be tested at different stages before it can be used. So her question is, has the COVID vaccine been tested this long for use, or it isn't necessary for it to take that long? It's, yeah. So let's just, Rebecca, just give a hopefully simple, direct answer to this question. Not too long, please. If you can unmute yourself. Thank you.
All right, thank you, Mona Lisa, for that question. It's actually a good question.
Mhm.
It takes a while, naturally, all the time. People see that it takes quite a long time for drugs and vaccines. And a lot of the reasons is because it takes them time to produce these drugs. It also takes a lot of bureaucracy to be able to put in their papers, I want to do this, and then they go through it and so on. And then the information hoarding, as in, I have this, I want to protect myself from another person making the money or the things from it. And then sometimes they have a lot of apathy, new drugs or new things, volunteers who will come out and then be tested for these things. And then there's, of course, like I said, funding challenge. But with this COVID, it's like the world realized, everybody worked together. The world realized the danger. People were dying from COVID and everybody cooperated. For instance, China, you know how it can be, but China released the gene sequence for it. Everybody go out there and do what you can. And so
yes,
everybody had
sorry,
the gene
Dr. Rebecca.
Okay.
Just to interrupt there, yes.
Technical?
Yeah,
okay.
No, just 1 second. Yes, just to talk about the background, that the research into the mRNA had been going on for quite a long time, over 5
exactly.
decades.
Very good.
So
yes.
we just want to narrow it down to that kind of information so that
So it didn't take time. It didn't take too much time because, like you just pointed out, a lot of background has already been, like the mRNA research has already been going on behind all this while, so that what they did is, once the gene sequence for the disease-causing agent, which is the SARS-CoV-2, was available, they now utilized it to make this. And a lot of people volunteered as in, let's try and stop this together. So everybody worked together, and that is how everything came out faster. So that's the reason.
Yeah, so essentially the background work had been going on. The journey of mRNA is, 5 decades, like you said. Yes, so the foundation was already in place.
Laid.
Yeah, absolutely. Thank you very much for that. Okay, do we have any more questions from the audience here or on YouTube or on Facebook? We've got these top researchers here. Now is the time to grab them before they go. Once they're back in the lab, you might not get to see them again for a while. Any more questions? Okay, Archer. Okay, oh, now they're coming thick and fast. 1 second. So Archer is asking, why aren't we seeing more human trials for medicinal plants as we see from orthodox medicines in chronic diseases like diabetes? Prof, over to you.
I had a feeling that one was coming.
Absolutely, yes.
Yes. I think that one of the major reasons we're having problems with clinical trials is they're incredibly expensive. And someone has said in Nigeria, we manage everything. And these are the corners you can cut in drug discovery. One of the papers that I wrote recently, I looked at medicinal plants that have been used to treat diabetes. People keep isolating and finding what is in this plant. The gap now is, how do we move this to clinical trials? How do we move it to a place where we can now confidently say that these plants or the substances isolated from them can be used to treat diabetes? The major problem is funding, I think, in my own opinion. And I think that as we go out more to look for better funds and create better awareness, we may be able to overcome this hurdle. I know that in places like China, where traditional medicine is entrenched into their system, they are encountering less hurdles over there. So this is one of the things we can look at. I started my career with just trying to find out whether it works. I had to move to, now I know it works. Why is it working? And now we have to move to, now we know how it works. What can we give back? What drug, what compound can we give back? And there are a lot of groups in Nigeria that are now looking at that. I work with a group from, incidentally here in Edo State, and they are trying to look at the first clinical trial for a medicinal plant. And if they can do that, that's wonderful. So that's basically the answer to that. Funding is a major issue.
Okay. All righty. Prof, I'll come back to you. Question for Ifeoluwa. What other, just 1 second. What other parasites can be found on unwashed fruit and veg? Oh, unmute yourself, please. Yeah. Is she frozen? No. Go ahead and unmute.
She needs to unmute.
Yeah, I've asked her to unmute. I don't know what's happening. Maybe the system is slow to react. All right, I'll come back to you, Ifeoluwa. Prof, question about gene therapy.
Yes.
Can gene replacement therapy be potent for type 1 diabetes, and is this achievable in Nigeria?
Okay. Diabetes is a multifactorial disease, but that means that it is caused by several factors. Genetic predisposition is just one of those factors. So gene therapy will work better for a disease where you have a single mutation. That is just one single issue. So it will work better there. For a multifactorial disease like diabetes, you would be using other approaches, or at least I think that would be the best way to go about it. If we're talking about sickle cell anemia, which is a single mutation, gene therapy would be more straightforward. But please note that these are therapies that are still being perfected, and it's not something that you find commonplace, so to speak.
Okay.
Thank you.
Thank you. Another question for you. Does consuming excess sugar necessarily cause diabetes?
Okay. Well, consuming excess sugar can predispose to obesity. It can alter the way your body produces and responds to insulin. On the short term, you may not see effects, but on the long term, it may bring about a dysregulation, so to speak, of the way your body responds to insulin. So let's just put it this way: a person who does not consume too much sugar has better insulin regulation, and than a person who does that. So, and diabetes, the key defect in diabetes is the insulin itself. Either it is absolutely lacking, that is not present at all, or your body is not responding properly to it. So I would advise anybody, try not to consume too much sugar. It's not good in any way.
Thank you. Thank you. All righty. Ifeoluwa, yes.
Yeah.
So what other parasites that's fine. What other parasites can be found on unwashed fruit and veg?
Okay, several studies have been conducted on fruit and vegetable, and some of the parasites that can be seen are Ascaris. We have the Entamoeba histolytica. We have the Taenia species, which is Taenia solium, Taenia saginata. We have the Giardia. We have the Cyclospora, and so many more.
Thank you for that, Thank you, Ifeoluwa. So Dr. Rebecca.
Yes.
Oh, good. Question for you. Please explain what mRNA is.
Okay. So
brief answers, please. Because of time.
mRNA is a little complex, but not just briefly.
You have to break it down
People
and keep it short. Thank you.
People ask a lot about that. They wonder if it changes their genes or something. So messenger RNA, simply used as with vaccine, is just that this the mRNA is used as it is used like a vector. It is used as to carry. The RNA is used to carry, to produce. It's given to the person, and the person's own protein, that is what does the job. And the person's own system is what utilizes the gene, which, a segment of the gene for maybe this SARS-CoV-2, depending on the virus, this SARS-CoV-2. So mRNA, I don't know how to simply make it now. So mRNA has to do with knowing the gene and utilizing it for your own system to make whatever it is it wants to make. So nothing foreign per se, nothing that can give you disease is used. So this system was discovered. Work has gone on it for over decades. So what it does, so instead of now giving like certain types of the disease, which can cause some form of disease, this generally is safer, gives you a segment of the gene which codes for the disease-causing agent, and then once it gets into your system now does the job of producing the protein that simulates the particular virus or gene it wants to vaccinate against. So it has been ongoing for a very long time. It's a technology that is out there, stabilized, has been used for many vaccines, including malaria vaccines that are out there, and has been found to be very, very successful and has been used for this COVID. I don't know. So
thank you for that, and thank you for keeping it short. So question for everyone, all the speakers. Well, two questions really. How do I decide which area of science to focus on for research? That's the first one. And the second one is, what can I do as a secondary school student who is interested in science? So let me see who wants to go first. Ifeoluwa, you can go first.
Sorry, please, I'm trying to comprehend the questions. I didn't get the questions very well.
So second question, what can I do as a secondary school student who is interested in science? And how do I decide which area of science to focus on for research? Anyone can go.
All right, let me go.
All righty.
So
remember, short and sweet, please.
Using myself as an example, I think it's all about, it takes time to find your passion. So as a secondary school student, if you like the sciences, then that is your niche. Some people can be actually gifted, as in you like the sciences, you like the arts. But if your interest is with the sciences more, you are held spellbound by them, you're curious, you're passionate about it, then follow your heart. That could be your lead. And then you go towards it. Then in deciding which area of science to go through, that is the kind of decision you make mostly during or after your undergraduate studies. When everything is laid out for you there, then the one that holds your interest, that you can study and read about and dream about, the one that keeps you awake, that is the one to go for. Passion is what I think it is.
Awesome, thank you. Who wants to go next?
I think I'll go next.
All right.
I think for students, what I would advise is just focus on what excites you. I'm talking not just secondary school students, I'm talking about all students. I have, the past 6 years, I have been focusing on mentoring, not just postgraduate students, but early career academics. And I see they thrive more when they do what interests them. So you don't have to push them. You practically, it's like giving them a kid a candy and they just run with it, so to speak. So if it interests you, if you love drawing and you want to force yourself to do something else, it becomes boring for you. But if you love science and you want to do something else, you'll never be fulfilled. I don't know, maybe it's me, but I'm excited about science. I love science because I love the fact that things that you think are inexplicable can be explained by this simple thing. So passion is an important thing. And if you can find a mentor, I would really advise you to get one, because mentors can take you places. Thank you.
Thank you. All right, Ifeoluwa, do you want to go next?
Yes, so how do you decide which area of science do you want to focus your research on? Like one of the speakers said earlier, I think when you can do this is probably after your first degree, because I think your first degree just gives you the whole picture. So after your first degree, you will be able to now know that, yes, this is what I want to focus my research on, or this is what I want my research focus to be. And also, as a secondary school student, I think that in junior secondary school 3, they do all subjects. So then you will know if your strength is in the sciences or not. And if your strength is in the sciences, which part of the science excites you? Where do you have passion for? And I think that also helps.
Fabulous. Thanks, Dr. Moyinoluwa.
Okay, I would like to say that everybody has a style of learning. So as individuals, we have to know our learning style. One, I'm a kinesthetic learner. I'm hands-on. That is why I went for, my dad told me something. He said, you can't do pure and applied biology. You had to do something else. And I was like, why? Counsel. Yes, there's a place of counsel and know your learning style. Then my sister is a doctor and she told me, biochemistry is a bit abstract. Microbiology is a bit what you can see. And I like to see what I'm doing. Simple and short. So I want to see what I'm doing. And that's micro for me. Microbiology is hands-on. I'm seeing what I'm doing. My bacteria can do this. I can see it. It can produce this. I'm seeing it. So everybody has that thing, that the kind of learner they are. So if you know the kind of learner you are, you'll be able to fit into where you find yourself, whatever part of science you are. There are some parts of sciences that are abstract. Some are more hands-on and all that. Then also, where you research, as Prof said, it's something that interests you. And there's also, as she said, mentorship. There's a place of guidance. People always teach people. People tell, why not try this? And I did not like cellulose from the beginning. My supervisor practically forced it on me at master's level. And I'm like, she was like, did you work with waste in your undergraduates? Why not continue with waste and see what you can do? And I found myself doing cellulose, and I found myself liking it, and I found myself doing research, even going from polymers, material science, and analyzing, characterizing. It was not what I thought of. And I found myself liking it. That's the place of mentorship and guidance. There are some people that just push you to it and you must do it, and you find out you like it. So we should also listen to people that will guide you and mentor you.
Thank you. Thank you. Dr. Oyelowo?
Okay, I'll just follow the line of these speakers. For a secondary school, I think you need to know yourself. What do you really like? Like the last speaker said, there are different arms of science: maths, physics, chemistry, biology. Physics was not it for me. So I knew that all that calculation for the maths was not for me. I prefer to read. I can read and understand what I'm reading. And then you tell me something, and then in practicals, I can see it, not telling me about Pythagoras theorem. Some people can do that, fine. So you need to know, your ability. And there's also the place of talent. Some people have cross talents. For instance, you are in science, you're artistic. It doesn't mean it won't work. For instance, there's what is called medical journalism. I do a lot of writing. So I like writing because I had an issue at the time. I wasn't sure if I wanted to do art or science because I love writing, I love drama. I have applied it to my science. When I teach, some of my students say I'm dramatic, but they remember what I've said. That's the most important thing. So I think there's a place of knowing yourself, discovering yourself, and be sincere with yourself. And also to discover the area of research that you will like. I think that should be after the undergraduate, when you really know, these are the available options. This is what I like. This is what won't stress me. That way you'll be able to fulfill your destiny, if you like.
Thank you. Amazing. Thank you very much for those answers, and I hope that has inspired someone or some people, or lots of people, to get into science. Now, I am going to hand over to the team lead, Dr. Orode, for some final comments.
Thank you all so much. So I've enjoyed being behind the scenes and trying to handle some of our tech. So I'd just like to say a very big thank you to everyone who has stayed this long to listen to our speakers. It's been a pleasure being able to bring Soapbox Science 2021 again to Lagos for the third time. Fabulous work and support from our sponsors, Encapsulate Healthcare Solutions, Dish Media. You should really go check out their app as well, keep you engaged with science that's happening in Nigeria. And they've been in the room, and I just want to say thank you for that. You can see our speakers in their lab coats all branded with their names and the Soapbox Science logo. That has continually been supported over the past 2 years by Elias and associates. Without our sponsors, it would be very difficult to host an event such as Soapbox Science, which is completely voluntary. So all the team, everything that is done is on a voluntary basis. We often start off without a budget. We don't usually have anything, and it's completely reliant on contributions from sponsors to be able to host it either online or in person. So I'm really grateful for that. And last but not the least is our wonderful speakers who take out the time to come and engage with you, share their science, this fabulous work that is ongoing in Nigeria. I often find that it's a shame oftentimes when scientists are not able to engage with the public. And I think there's a very great need for that in our country. So I'd just like to say thank you so much to all our speakers for sharing their time with us, sharing their time with you, and sharing some of the things that they're able to do in the lab. I have mentioned in the chat that if you signed up for, if you noted during your registration on Eventbrite that you'd like to attend a Careers in STEM seminar, you can hear a few of our speakers saying they're mentors. So we might be approaching some of them to come and engage with you and inform you about the different types of careers that are available in STEM across science, technology, engineering, maths, and medicine. And yes, mentors, I completely echo that. Mentoring is a very great tool in your career as you try and decide. When I was trying to decide, do I go into maths or biology, and how I was able to make that choice, microbiology or chemistry, I find myself doing both at the same time. So yeah, and I love that comment was it Dr. Oyelowo talking about medical journalism? It's not something a lot of people know. Science writing. So if you're still in the room, do take a note and explore those, because they're not very popular oftentimes. You have scientists work, I use the word scientists very generally. Scientists work across different sectors. So if you're interested in that, please look in the chats and drop us an email. There was one last question. I don't want to leave this member of the audience hanging. She's on Facebook. Abigail was asking about Since COVID-19 is said to have many variants, we're taking a vaccine necessarily protect one from the so many variants and even the ones yet to be identified. Abigail, I'd like to quickly draw your attention to the influenza vaccine. Influenza might not be so endemic as it is here in the United Kingdom. Yes, as we can imagine with every disease, like I think it was Dr. Rebecca mentioned, you will have variants. She talked about some mutations occurring with different bacteria because they're trying to get used to their environment. And one of the things we have with influenza is another key area in science called surveillance. Surveillance is very, very important. And you often have that new vaccines will be developed to check which variants are being so much in the environment and which vaccine we need to use to target that prevailing variant. So I hope that kind of gives you a simple idea into that area. You might want to go and read more about it. If you're still interested, please drop us an email and we'll send you the relevant information. So without much ado, I'd just like to say thank you. We bring the event to a close. Speakers, please don't leave. We just have a final debrief. Thank you so much for your time, attendees. It's been a pleasure hosting you. God bless you, and hopefully see you next time. Bye.